Okay, in this short video, we're still continuing on connective tissue. We're going to be talking about liquid connective tissue. And one of the important liquid connective tissues is called blood. When you have blood drawn, blood is taken from a vein. That's why it's called a venipuncture. It's taken from a vein. It is then spun and it goes through the centrifuge, and the different elements within blood separates. And the lighter substance is at top, and then the heavier substances sink to the bottom. So the portion up here is 55% of the blood is plasma, and then 45% are cells. Now remember, connective tissue is when you have cells that are not so closely packed together with lots of extracellular matrix. The plasma is the extracellular matrix. Okay, so let me get my pen here. So the plasma is the extracellular matrix. When you have more matrix, when you have lots of matrix and less cells, that's what really connective tissue is about. So when we look at, when we look at the blood, 55% is plasma, and plasma is made up of proteins, a large portion of water, right? 92% water, and then other solutes. The formed elements, which are the cells that sink to the bottom, that makes up the 45%, that's where we have white blood cells, red blood cells, and platelets. So we want to be able to differentiate what does a platelet look like, what are the different white blood cells, and what are the different red blood cells. So that's what we're going to look at now. So red blood cells are kind of unique, right? They look like, under a microscope, the outer part is thicker, and then the inner part is thin. And you can see on a cross section, the periphery of it, this outer portion, looks a lot thicker, right? Here, this part looks thicker. And then this inner portion, the inside of the donut, is thinner, right? And so light can pass through the center of a red blood cell. So it typically gives the inner part of the donut a lighter appearance. So we call the red blood cell, the RBC, it's like a biconcave shaped disc. And the name for the red blood cell is called an erythrocyte. So all of these on the left-hand side under a microscope, every single one of these large cells that we see is a red blood cell. We could see that the inner part of it is much lighter, right? All of these have a central area where it's lighter, and that's because of this biconcave disc, right? So think of it as a donut. Those are red blood cells, or erythrocytes. Now we have white blood cells. Let me just see if I can go back here and show you the breakdown of white blood cells. Let me see if I have them here. If not, I'll draw them out. No big deal. So red blood cells are known as erythrocytes, and these are the most abundant blood cells, and their main job is really to carry hemoglobin. And in hemoglobin, we have iron. Fe is iron. And what's going to bind to this iron is going to be oxygen. It's going to be drawn to that. So those are the red blood cells. Now, white blood cells are called leukocytes. And there are 5 main types of leukocytes. Now we have granular cytes. I'll put a G for granular. And then we have agranular. Granular and agranular. What I mean by granular is, let's say here's a cell, and let's say here's a nucleus. When you have a bunch of sandy-like granular substances in the cytoplasm and it looks grainy, then we say that's granular. When you have a cell that has a nucleus in the center and the cytoplasm is clear, then that's agranular. A means without. So the three type of granular cells, I call them BEN, B, E, N. The B is for basophil, the E is for eosinophil, and the N is for neutrophil. You don't have to write these because they're up in the PowerPoint. So basophil, eosinophil, and neutrophil. Basophil, eosinophil, and neutrophil. They all end in the word phil, P-H-I-L. And phil means to like philos, for friend of, philosophy, the love or friend of wisdom. So basophil, when you have a basophil, that takes up a basic stain. When you have an eosinophil, that takes up an acidic stain. And when you have a neutrophil, that's neutral, not necessarily basic, not necessarily acidic. So it's neutral. So you have the basophil, eosinophil, and neutrophil. Those are the three types of granular sites. And then we have a monocyte, and we have a lymphocyte. Those are agranular. Monocytes and lymphocytes don't have any granules, okay? And then the third type of formed element that we have, they're called platelets. They're also called thrombocytes with a T. Let's change that. Thrombocytes, they're involved in clotting. Okay, platelets or thrombocytes, they're involved with clotting. And this is why many people take baby aspirins, because baby aspirin blocks the function of platelets, so your blood thins and it doesn't clot. So now we're going to look at these 5 cells, the basophil, eosinophil, neutrophil, those 3 granular sites, and then we'll show you the monocyte and lymphocyte, which are the agranular sites. So when we look at this, and we look at this, and we look at these 3, notice how within the cytoplasm we see granules. So these are granular sites. These are granular sites. When we look at a basophil, let's look at the neutrophil first. That's the first one in order. Surrounding that, we have a bunch of red blood cells. We see that the central part, we call it hypochromic, light in color, hypochromic, less color. And then we have this large cell here, and this large cell has a nucleus. And the nucleus here looks like it has 1, 2, 3, 4, maybe 5 heads to it. So when a neutrophil can have anywhere from 3 to 5 heads of the nucleus. So in this model, we have 1, 2, 3. And then we have these granules, which look a little bit red, but they're small, right? They're very, very grainy. And that looks a little bit different than the eosinophil, which also has a nucleus, but this nucleus is what we call bilobed. It only has 2 lobes. This is one lobe here, and this is the other lobe here. So when you have 2 lobes to the nucleus, and again, you have red granules and they're large, that's an eosinophil. The eosinophil used to be referred to as an acidophil. They used to call it an acidophil because it takes up an acidic type of stain, which turns this red, but these are large red granules. Now, these granules can degranulate. They break and they release whatever is within them. So these granules also have substances within them, and then they break and they release what's within them. Neutrophils are like your first responder. That's your first responder. So when you have a bacterial infection, when you have a bacteria, your neutrophils are the first line of defense, right? It's like your first call to action. Neutrophils come. These neutrophils are going to open up and release their strong digestive enzymes to try and destroy that bacteria. So when you do blood work, if you have a high neutrophil count, it means that you're fighting a bacterial infection. When you have a high eosinophil count, it means that you have allergies. There's some sort of allergy, or you have a parasite or a parasitic infection. That's when the eosinophils are high. Then we have a basophil, and the basophil also has large granules, and the nucleus, everything within this basophil is darkly stained. So when the nucleus kind of gets obscured by the dark stained granules and it's hard to see, it says the nucleus generally cannot be seen through the dense blue stained granules in the cytoplasm. So you see the basophil, blue, dark blue, black, very dark staining. That's your basophil. Okay. And basophils, they can release histamine. They can release histamine in a similar fashion to another type of cell called mast cells. Mast, M-A-S, also release histamine. So basophil, darkly stained. Eosinophil, bilobed, large red stained granules. And then the neutrophil, 3 to 5 lobes to the nucleus, small red stained granules here. Those are the 3 granular sites. Now, the 2 agranular sites are called monocyte and lymphocyte. The monocyte is a large cell, it's agranular, and it's known to have this kidney bean-shaped nucleus. Okay, that's the key, the kidney bean-shaped nucleus. And it's pretty large in relationship to these red blood cells, agranular. Monocytes are always in circulation in your blood, and when they move into tissues that are damaged, it's called a macrophage. A macrophage. Or a phagocyte. C-Y-T-E. A phagocyte. So monocytes become macrophages, and macrophages are just large eating cells, and that's what a phagocyte does, or phagocytosis. It means cell eating or cell engulfing. It's going to find debris within your tissues and destroy it. So monocytes are in your blood vessels, and when it finds some sort of issue going on in your tissues, it leaves the blood vessels, goes into the tissues, and does its work. Depending on where that monocyte goes into, that macrophage can change its name. So when the monocyte goes into, let's say, the liver, it's called a Kupffer cell. When that monocyte is trying to break down bad cholesterol, it's called a foam cell. When the monocyte goes into the brain and the nervous system to engulf debris, it's called a microglial cell. So depending on the tissues they go into, they can alter and change their name. A lymphocyte is smaller than a monocyte, and one of the key features is look how big that nucleus is, right? The nucleus almost occupies the same amount of space as the cell itself. So it has a largely, it's round, it's slightly larger than the red blood cell, right? Slightly larger. It's got a round nucleus, very symmetrical, but it's got very little cytoplasm. That's because the nucleus is so big. Okay. And then this is a platelet. A platelet is involved with clotting mechanism. It creates a thrombosis. So let's take a look at this plaque and let's see if we can identify some of these structures. Okay, so let's look at number 1, which is a red blood cell or an erythrocyte. So here is number 1. That is a red blood cell. Let's look at number 2, a lymphocyte. So this one here is the cell that has the nucleus that's pretty round, very little cytoplasm. The nucleus is just as big as the cell. There's no granules, so that's a lymphocyte. Now that's a little bit different than number 3, a monocyte. Number 3 is much larger, right? It's the biggest cell that we have, and it has that kidney bean-shaped nucleus. So that's a monocyte. Remember, monocytes are in blood, and they become a macrophage. I'll do MPH, a macrophage. Where's our number 4? Let's look at our neutrophil. This little typo there, take out that R and just call it a neutrophil. Remember, they all end in phil, which means they like something. The basophil is the dark staining one. The eosinophil is the red one. And the neutrophil has a little combination of both. So the neutrophil, number 4, if I were to ask that, I would point to this one here. Why? Because it has 1, 2, 3, 4 heads to the nucleus. Okay, or I'd ask something like this one here, also 4 heads to the nucleus. Or I'd ask this one here that has 3 heads to the nucleus. So once we see 3 heads, we know that it's a neutrophil. In contrast to number 5, which is typically bilobed. So if I were to ask an eosinophil, I would ask this one, which clearly has 1, 2 lobes, or I'd ask this one here, which clearly has 1 or 2 lobes. I would ask you something very clear and distinct for the eosinophil. Whereas the basophil, number 6, that's the one that has the dark staining, that dark blue granules. And then platelets are number 7. All of these things here, these are platelets. These are all platelets for clotting. And then the green substance, we see number 8 here, all of this matrix, all this green yellow stuff between all of the cells, that's your plasma. Okay, that's the plasma. So if we were to look at these again, now let's look at what they look like, not on the slide, but here. These are, this one right here, if we look at the nucleus, 1, 2, 3, maybe 4 heads to the nucleus, so that's a neutrophil. An eosinophil only has 2 lobes. So here's 1, 2, and it has red staining granules all around it. That's the eosinophil. If we look at a basophil, if we look at a monocyte, this one's agranular, it's large, this has the kidney bean shaped, that's your monocyte. This one here is very grainy, hard to see if there's a nucleus, but it's granular. So the rule is if you have trouble seeing the nucleus and it's very grainy, that's your basophil. This one again, 1, 2, 3, this one's got 4 heads to the nucleus. Hard to see the granules, but you could easily see the nucleus and use that as a rule. 1, 2, 3, 4, that's a neutrophil. This one, we have a nucleus that's just as big as the cell. Very little cytoplasm, that's the lymphocyte. So now let's see if we can name these cells. Okay, we don't care about the RBCs. We know that red blood cells are in all of these. Which is the largest one? Let's see, which is this large cell that has this kidney bean-shaped nucleus? We'll say, I'm going to say that that's a monocyte. Hopefully you agree with me. Which is this cell that's agranular, has a nucleus that's just as big as the cell, very little cytoplasm. I'm going to say that this one's a lymphocyte, lympho. Hopefully you agree with me. Okay, let's look at this one here that has clear red staining granules. This is 1, 2 heads to the nucleus, very, very red. I'm going to say 4 is an eosinophil. Number 5, everything is dark, dark blue, 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 very, very dark. That's the key for B and dark, basophil. And then here we have 1, 2, 3, 4 heads to that nucleus. I'm going to say that's a neutrophil. Let's see what we have. So monocyte, lymphocyte, 1, 2, 3, 4 to 5 heads to the nucleus, neutrophil, 1, 2 heads to the nucleus, bright, bright red cytoplasm with granules. That's your eosinophil, which used to be called acidophil. And then the basophil, which takes up that dark blue, purplish basic stain. Everything stains dark. That's our basophil. Okay, I know someone had a